synthetic biology techniques to produce hydrogen gas as a renewable fuel alternative. Global warming and climate change are big threats to our ecosystems, caused by the way we source and consume our energy. To address the issue of climate change, various renewable energy options have been considered, but none have been able to fully replace the fossil fuels damaging our atmosphere. One such example is hydrogen energy. Our project aims to utilise living cells to produce hydrogen gas for our consumption and to lessen the burden of energy production in the non-renewable sectors. Nature already has ways of producing hydrogen gas. In fact, many microbes produce it all the time. Using synthetic biology, we aim to copy the DNA sequence of one such organism, the algae called Chlamydomonas reinhardti, and insert it into E. coli bacteria. This will allow us to mass produce hydrogen gas on a much larger scale than naturally occurs in algae, in an effort to curb our reliance on fossil fuels. So what is synthetic biology? It is a relatively new field where engineering techniques are applied to biology to program organisms to have new functions. This involves the creation of standardized parts of synthetically made DNA coding for specific functions called biobricks. These work much like Lego bricks. And individual parts that we can join to build a DNA sequence and insert into a bacterial host to express our desired function. The great thing about these biobricks is that we now have a huge collection of standard parts that have been submitted to a registry that is open sourced. This registry is run by iGEM, the International Genetically Engineered Machine, which is a competition where university teams from all around the world design and submit their own biobricks to address various issues. We are part of this year's Macquarie's iGEM team. Macquarie is the longest running Australian team and has won 3 gold medals over the past 3 years, so we have some big shoes to fill. There are many applications for synthetic biology. It can be used to make pharmaceutical medicines, finding ways to combat antibiotic resistance, for food and agriculture. It can also be used to find alternative fuel sources, which is what our project is based on. As a fuel source, hydrogen energy has a lot of potential. Its combustion produces no pollution or greenhouse gases. However, these advantages have little meaning when we consider the costs of extracting hydrogen. Hydrogen gas is not found in abundance and is expensive to synthesize and extract. As you can see here, 96% of our hydrogen is produced from non-renewable sources, such as coal, oil, and natural gas. Therefore, hydrogen gas as an energy source is not desired by the majority of industries. There have been improvements in this technology, however, which limit these detrimental impacts. This has increased the interest for hydrogen fuel in the Australian market, with groups such as the CSIRO predicting hydrogen to be our next biggest export. Hyundai has been trialling their hydrogen car and aiming to release them in 2018. In fact, right here in South Australia, the government is getting in on the action and recently made an announcement that they aim to release a new fleet of buses within the next 2 years, completely run on locally sourced hydrogen energy. In anticipation of this future, our team aims to overcome the major limitations of hydrogen gas production, which are 1, the way in which it is sourced, and 2, the way in which it is stored. So how do we plan on doing this in a clean way? The first issue is addressed by photosynthesis, a natural process by which sunlight is converted to energy for plants, and we are looking to use synthetic biology to capitalize on this process.
So first, discussing mostly on the synthetic biology aspect, I'll first go over what happens in cells naturally. Now, both E. coli and Chlamydomonas both produce hydrogen gas, but they're all in negligible amounts. This is done through a complex called HydroGEM, which uses electrons and protons to facilitate the hydrogen reaction. A lot of the electrons and protons can be sourced from any place in the organism. Even in our bodies, we're always trying to source these items. In algae, such as Chlamydomonas reinhardti, the most abundant source can be found through the systems that control photosynthesis, an example being photosystem 2. In the long-term project, we do hope to use photosystem 2 into our E. coli cell. However, presently we are using something called ferredoxin, which we're using as a source of electrons and protons. So we aim to incorporate this system in the long run, but what we've done so far is, in this little schematic diagram of our cell, you can see the genes that we have used and how the cell is going to be transcribing them and in what order. Okay, so after introducing our E. coli to the new DNA, we allowed it to incubate in optimal conditions and then screened them for growth of just the selected DNA that we wanted. This cell here shows that we were quite successful or just very, very lucky. Take your pick. What happened was that we ended up with quite a lot of colonies. So generally what tends to happen is when you're doing colony works on plates such as these, you'll find scattered colonies here and there, and sometimes, and this happened to us at one point, you'll spend an entire week working on making the cells competent, trying to make sure everything's going to work, to get 0 cells. Thankfully, we have a really nice image showing that it didn't happen. Now these cells tend to be turned on by glucose, which is what we did. So we added glucose, and it took approximately 2 hours for the cells to start producing hydrogen, which is fairly quick. You can see on this graph, the very first slope here, that's when we initially started to produce hydrogen, and then we started off having peak productions that we can see here. This peak production of hydrogen gas lasted for more than 24 hours, which means that we do have a quick way of producing hydrogen gas for renewable energy and combustion. However, after the 24 hours, we can see a decline in our hydrogen gas production, and after 7 hours, the production ceased. This may be because of the fact that we did not add any more glucose to further encourage the cells to produce. At this point, we were just trying to figure out, are our cells good enough? The next thing we did was have control cells. So these are standard E. coli cells found in everyday laboratories without our new chlamydomonas DNA attached to it. And over there on the graph, you can see that there is no hydrogen production, which shows that our synthetic biology tools were the ones making the difference and making hydrogen and hydrogen gas. The way we measured the hydrogen gas was through something called a Clark electrode. It is a pretty complicated system, but all you need to know is that the Clark electrode was measuring the electrons being used up in the reaction to produce the hydrogen gas.
Now, many great ideas in science unfortunately don't come to light if they are not well received or understood by the public, which is why education and engagement is such a vital part to science. As astronaut Chris Hadfield once said, science communication is vital to our quality of life. It helps everyone understand the problems that we face and shares the ideas and solutions that can improve life for us all. We have worked to close this communication gap between our research and the public by taking part in many outreach activities. We have worked on developing a video game aimed at high school and undergraduate students that teaches the fundamentals of synthetic biology in a fun and engaging manner. We have run workshops with both primary and secondary students to spur an appreciation of synthetic biology, renewable energy, and the aspects of our project. We have also been fortunate enough to be given the opportunity to promote our work at TEDx Youth Sydney, which was broadcasted globally, and to present at conferences such as the Synthetic Biology Australasian Conference and ACUR.
One other thing that we also did was we've been working on a prototype. So we mentioned earlier on that there are 2 limitations to hydrogen gas usage, the first one being production of the hydrogen gas, the second one being the storage. So what we did was we surveyed more than 150 potential consumers of our product, and based on their responses, we are currently in the process of designing an at-home prototype device which can use our cells to produce hydrogen gas. Through this project, we have success on both fronts with our E. coli cells, quickly and easily producing hydrogen gas cheaply for renewable energy purposes, and also in storing the hydrogen gas for at-home usage. None of this would have been possible without our sponsors and the support staff. And we would also like to thank, in particular, Dr. Louise Brown, Professor Robert Willows, and Dr. Paul Jashke, who've been guiding us throughout this entire process. If you do have any questions or would like to get in contact with anyone from our team, feel free to contact us through the social media. I tend to monitor that all the time. Yeah, that's all we've got on. Thank you so much.